The genetic code. Proteins are made by joining amino acids into long chains called polypeptides. Each polypeptide contains a combination of any or all of the 20 different amino acids. The properties of proteins are determined by the order in which different amino acids are joined together to produce polypeptides. How, you might wonder, can a particular order of nitrogenous bases in DNA and RNA molecules be translated into a particular order of amino acids in a polypeptide? The language of mRNA instructions is called the genetic code. As you know, RNA contains 4 different bases, A, U, C, and G. In effect, the code is written in a language that has only 4 letters. How can a code with just 4 letters carry instructions for 20 different amino acids? The genetic code is read 3 letters at a time, so that each word of the coded message is 3 bases long. Each 3-letter word in mRNA is known as a codon, as shown in figure 12-16. A codon consists of 3 consecutive nucleotides that specify a single amino acid that is to be added to the polypeptide. For example, consider the following RNA sequence: UCG, CAC, GGU. This sequence would be read 3 bases at a time as UCG, CAC, GGU. The codons represented the different amino acids are serine, histidine, and glycine. Because there are 4 different bases, there are 64 possible 3-base codons. Figure 12-17 shows all 64 possible codons of the genetic code. As you can see, some amino acids can be specified by more than one codon. For example, 6 different codons specify the amino acid leucine, and 6 others specify arginine. There is also one codon, AUG, that can either specify methionine or serve as the initiation or start codon for protein synthesis. Notice also that there are 3 stop codons that do not code for any amino acids. Stop codons act like the period at the end of a sentence. They signify the end of the polypeptide, which consists of many amino acids. Translation. The sequence of nucleotide bases in an mRNA molecule serves as instructions for the order in which amino acids should be joined together to produce a polypeptide. However, anyone who has tried to assemble a complex toy knows that instructions generally don't do the job themselves. They need something to read them and put them to use. In the cell, that something is a tiny factory called the ribosome. The decoding of an mRNA message into a polypeptide chain or protein is known as translation. Translation takes place on ribosomes. During translation, the cell uses information from messenger RNA to produce proteins. Refer to figure 12-18 as you read about translation. A. Before translation occurs, messenger RNA is transcribed from DNA in the nucleus and released into the cytoplasm. B. Translation begins when an mRNA molecule in the cytoplasm attaches to a ribosome. As each codon of the mRNA molecule moves through the ribosome, the proper amino acid is brought into the ribosome by tRNA. In the ribosome, the amino acid is transferred to the growing polypeptide chain. Each tRNA molecule carries only one kind of amino acid. For example, some tRNA molecules carry methionine, others carry arginine, and still others carry serine. In addition to an amino acid, each tRNA molecule has 3 unpaired bases. These bases, called the anticodon, are complementary to one mRNA codon. In the case of the tRNA molecule for methionine, the anticodon bases are UAC, which pair with the methionine codon AUG. The ribosome has a second binding site for a tRNA molecule for the next codon. If that next codon is UUC, a tRNA molecule with an AAG anticodon would fit against the mRNA molecule held in the ribosome. That second tRNA molecule would bring the amino acid phenylalanine into the ribosome. Like an assembly line worker who attaches one part to another, the ribosome forms a peptide bond between the first and second amino acid, methionine and phenylalanine. At the same time, the ribosome breaks the bond that held the first tRNA molecule to its amino acid and releases the tRNA molecule. The ribosome then moves to the third codon, where a tRNA molecule brings it the amino acid specified by the third codon. The polypeptide chain continues to grow until the ribosome reaches a stop codon on the mRNA molecule. When the ribosome reaches a stop codon, it releases the newly formed polypeptide and the mRNA molecule, completing the process of translation. The roles of RNA and DNA. You can compare the different roles played by DNA and RNA molecules in directing protein synthesis to the 2 types of plans used by builders. A master plan has all the information needed to construct a building, but builders never bring the valuable master plan to the building site where it might be damaged or lost. Instead, they prepare inexpensive, disposable copies of the master plan called blueprints. The master plan is safely stored in an office, and the blueprints are taken to the job site. Similarly, the cell uses the vital DNA master plan to prepare RNA blueprints. The DNA molecule remains within the safety of the nucleus, while RNA molecules go to the protein building site in the cytoplasm, the ribosome. Genes and proteins. Gregor Mendel might have been surprised to learn that most genes contain nothing more than instructions for assembling proteins, as shown in Figure 12-19. He might have asked what proteins could possibly have to do with the color of a flower, the shape of a leaf, a human blood type, or the sex of a newborn baby. The answer is that proteins have everything to do with these things. Remember that many proteins are enzymes, which catalyze and regulate chemical reactions. A gene that codes for an enzyme to produce pigment can control the color of a flower. Another gene produces an enzyme specialized for the production of red blood cell surface antigens. This molecule determines your blood type. Genes for certain proteins can regulate the rate and pattern of growth throughout an organism, controlling its size and shape. In short, proteins are microscopic tools, each specifically designed to build or operate a component of a living thing.